Genome Project was a huge project in which many methods have been established that later on were transformed into the Human Microbiome Project. And of course, the gut was the primary goal, the primary aim to discover, but very soon after, the skin was investigated. And it is probably only 10 years from today that we have gathered all the information about the cutaneous microbiome, the role it has in orchestrating and training the immune system, the role it has in governing wound healing, the role it has, but also in mounting inflammatory skin diseases such as atopic dermatitis. And I think what we learned is that the microbiome that we have on our skin is actually rather stable and not as temporary as one might have thought in the beginning. It is stable and it is part of our cutaneous barrier. So we used to think of our cutaneous barrier as being a physical barrier, a chemical barrier, pH 5. But now we understand that it's much more complex. It's actually several levels of cutaneous barriers that network together and that make up our complete barrier. And so the microbiome is actually the most outer part of these barriers, followed by the physical and chemical barrier, and then the innate immune barrier, followed by the adaptive immune barrier follow. And so we have to understand that these barriers, these levels interact tightly, that they regulate each other, and that a disturbance on the microbiome will have consequences on the other barriers, as well as a disturbance in the immune system will have a consequence on the microbial composition. So understanding these different levels, I think it is very important that once the barrier is disturbed, maybe on a genetic basis like filaggrin mutations, then we will have a disturbed microbiome, a dysbiosis. And if inflammation is mounted within the skin, we also will have dysbiosis. And maybe there's even primary causes of dysbiosis that will secondarily induce the reduction of the physical barrier as well as a deviation of the immune system. What we also learned is that the microbial system on our skin really trains the immune system. This is experimental data, but they show pretty directly that without our microbiome on the skin, we will not be able to mount effective anti-infectious immune responses. It was shown for Candida albicans infections. If you don't have commensals on the skin that train your immune system, you will be very susceptible to Candida infection and not be able to really mount an immune defense. For example, on the other hand, we understand that our immune tolerance, so the active tolerating of agents on our skin, is also trained by the commensals, by our microbial system on the skin. So both defense as well as tolerance is orchestrated through the microbes. And additional information tells us that wound healing is also very much dependent on very old pathways of innate immune triggering that follow commensals on our skin. And if something goes wrong, a lot goes wrong. And here I come back to atopic dermatitis, where we know that dysbiosis, the dysfunctioning of the microbiome, is one important part, as well as the barrier and the type 2 prone immune system with interleukin 4 and interleukin 13 being the dominant. We have data today showing that just the immune deviation towards type 2 immunity is enough to reduce the physical barrier and induce dysbiosis. We also know that reducing the barrier, on the other hand, will allow type 2 immunity to develop much easier and will also lead to dysbiosis on the skin. So there's 3 entry points into the disease actually. This is the microbiome, this is the barrier, being the physical barrier as we used to call the barrier of the skin, and the immune system.